So in this review, what I'd like to do is fill in this chart regarding the different classifications of biological macromolecules that we learned about in lecture and give you at least a quick and easy cheat sheet to kind of go off of. Now this isn't going to go in as great a depth as, say, we went into when we were reading through the book and going through the PowerPoint slides, but this would hopefully be a good starting point for you to use while you're studying for your exam. So first and foremost, let's start with the different classes. Recall that we learned about carbohydrates, we learned about lipids, proteins, and finally, nucleic acids. Also remember that all of these classes had distinct monomers except for lipids. Lipids do not have a defined set of monomers, because remember there are actually 3 subclasses of lipids that we looked at throughout the course of this lecture. So we're going to ignore lipids for now, and we're going to go ahead and start with carbohydrates. So remember that the monomers, or the single units of carbohydrates, are monosaccharides. And that's because carbohydrates are themselves a class of sugars. Mono means single, saccharide means sugar. So these are single sugars. And an example of one of the more popular monosaccharides that's commonly studied is glucose. If we take 2 of those monosaccharides and link them together through dehydration reactions, we can form a dimer that's called a disaccharide. And an example of a disaccharide is maltose. Furthermore, if we take more than 2 of these monosaccharides and link them together, we can eventually make what's known as a polysaccharide. And an example of a polysaccharide would be starch. Now, the linkage that is used ultimately to link these monosaccharides or disaccharides together is called a glycosidic linkage. Okay. Glycosidic, glyco meaning sugar, linkage. Okay. So, we've got monosaccharides, we've got disaccharides, and we've got polysaccharides. We link them together via a glycosidic linkage. Right? Now the functions of these molecules, and there are a couple. If we're talking about the mono or disaccharides, the function we're talking about is a ready supply of energy for the cell. If, however, we're talking about the polysaccharides, we're talking about either energy storage, or we're talking about structural support. So you'll remember that the cellulose and the chitin that we talked about, those were both utilized to provide the cell or structure therein with some form of structure. As far as additional information goes, one of the easy ways to know that you're always looking at a carbohydrate is to look for these 3 letters at the end of a word. Glucose, maltose, a form of starch called amylose, all end in OSE or OS. So that's how you know right off the bat that you're looking at carbohydrates. All right, let's talk lipids. So lipids didn't have monomers or even dimers, but we did technically have polymers that were assembled eventually to give us our 3 classifications of lipids. We had fats, we had phospholipids, and finally we had steroids. And at least in the fats, and to a degree in the phospholipids, what we saw in terms of a linkage was commonly called an ester linkage that was used to link those fatty acids to the glycerol molecule in either of these 2 instances. Now when we talk about functions of these molecules, going to abbreviate phospholipid as PL for now. Okay. Fats are going to serve the purpose of long-term energy storage. They're also going to serve to protect and insulate. Okay. Phospholipids are going to be used to make up membranes. And finally, steroids have various functions. Now, whether we're talking about fats, phospholipids, or steroids, those all have one unifying characteristic because we don't have unifying monomers. So the unifying characteristic of the class of lipids is all of them have a hydrophobic region. So all of them have a hydrophobic region. Okay? So 2 classes of macromolecules down, 2 to go. Let's talk proteins. The monomers or building blocks of proteins are called amino acids. Recall that these amino acids have very similar structures in 3 of the atoms or functional groups surrounding the alpha carbon, but they do have that distinct R group that diversifies them so that we can categorize them as either polar, nonpolar, or acidic or basic. So we're going to link these 2 or more amino acids together, and if we link 2 of them together, we're going to be left with a molecule we call a dipeptide. Whereas if we link multiple ones together, we're going to be generating a polypeptide. And we do this through a distinct linkage that is called a peptide bond. So we've linked amino acids together to form a dipeptide or a polypeptide. And assuming that that polypeptide folds into that tertiary or quaternary structure that we discussed today, then we're going to generate these molecules that have a variety of functions. And that variety of function can include things like receptor proteins, antibodies, we also have enzymes, we have structural proteins, and we have many more. Okay? There are contractile proteins, which you learned about. There are energy storage proteins. So there are a variety of different types of proteins that you can find in that section in your book. For additional info in regards to all of this, just recall that these must fold to be functional. All right, and finally, last class of biological macromolecules, nucleic acids. The monomers of these are going to be called nucleotides, and these nucleotides are going to be built out of 3 subunits: a phosphate group, a sugar, and a nitrogenous base. If we link 2 of those nucleotides together, we get a dinucleotide. And if we link 3 of them or more together, then we have a polynucleotide. All right? And the linkage that occurs to give us these di or polynucleotides is called a phosphodiester bond. So we get a phosphodiester bond. The functions of these molecules can depend on whether or not they're DNA or RNA. So DNA is going to house genes, and it can also allow for its own replication. DNA also allows us to have a blueprint through which we can make RNA, because RNA essentially is going to carry a message to be converted to protein. So the message in the RNA ultimately is going to be converted into protein, and that message is housed somewhere in the DNA. Now, DNA doesn't just house the genes that are used to make RNA. DNA can also be used as a template to replicate itself. A couple of other things worth noting. In DNA, the sugar that you're going to have is called deoxyribose. And the bases are going to be adenine, which pairs with thymine through 2 hydrogen bonds, and cytosine, which pairs with guanine through 3 hydrogen bonds. In RNA, you're going to have a sugar called ribose, and the nitrogenous bases you're going to have are going to be called adenine, which pairs with uracil through 2 hydrogen bonds, and cytosine, which pairs with guanine through 3 hydrogen bonds. Okay? So this is our cheat sheet chart to go through the different classifications of biological macromolecules.